All right, let's do bulk transport. This is the third and final part to lesson 9, which is all about diffusion. Part 1 was passive transport. Part 2 is active transport. And part 3 is going to be bulk transport. Do any of you go to Costco or Sam's to go shopping? Anybody? 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 And have you ever been to Costco or Sam's to go shopping? Okay. Well, if you want to get just one box of cereal, go to Publix. You want to get 5 boxes of cereal all in one giant box, go to Sam's or Costco. You want a little jar of mayonnaise, go to Publix. If you want like a barrel of mayonnaise, go to Costco. They sell that stuff for restaurants because it's just so big and they have a lot of customers. They need a lot of supplies. Well, there are some things that the cell needs or wants to get rid of or need to leave the cell that are way too big to just diffuse through those protein channels. Those protein channels are meant for ions and little molecules. But we're talking about macromolecules. Macro means big. And so they're far too big to just slide through a portal or slide through a channel or a pump to get from one side of the cell membrane to the other. So you have to go through a process of diffusion called bulk transport. And this type of diffusion is an active transport process. Because it's active transport, remember that it requires energy. And what is the most common form of energy for a cell? Adenosine triphosphate. Keep in mind that there is such thing as GTP, guanosine triphosphate. But ATP is more common, is what we generally deal with. When we call the mitochondria the powerhouse of the cell, I've heard that since 6th grade probably, it makes ATP, which is energy. So the deal here is large molecules, and we're talking like macromolecules, proteins, lipids, carbs. Large molecules are transported using vesicles. I think it would be important to rehash what a vesicle actually is. A vesicle is a sac of membrane meant to transport molecules into, out of, or around a cell. So, guys, think of a vesicle like your backpack, your lunchbox, a garbage bag, Tupperware, a container. It's just meant to move things around. If you're going to put a bag of Doritos in your lunchbox, you're not going to open up the bag and empty out all the chips in your lunchbox and just open up your lunchbox and boom, you got a big mess. You keep it inside of a container because it's easier to transport. That's what vesicles are basically designed to be. And keep in mind that their membranes are identical to the membranes of the cell membrane and organelles. All right. So there's going to be 2 major types of bulk transport we're going to talk about, and there's going to be a few examples of those 2 major types. The first of the 2 major types is going to be endocytosis. Today you're going to get a lot of Latin, so I think it would just behoove us to translate it, because throughout this year, you're undoubtedly going to see words that you do not know what they mean, but you can translate them based on your knowledge of the Latin affixes and figure out what they stand for. So if you look at this, you're like, I have no idea what endocytosis is. Well, what does endo mean? Inside or within. What does cyto mean? Cell. Does anybody know what osis means? It means process. You'll get that when you get to the O's on your Latin quizzes. So this is an inside cell process. You're bringing things inside the cell. This is all about bringing molecules from the outside. What do you call the outside of the cell? What is that area called? Beyond the cell. It's like extraterrestrial is beyond the Earth. What is beyond the cell? Is this Mina? Same thing. Extracellular space, extracellular matrix, pretty much the same thing. You go from the extracellular matrix or the extracellular space to the inside of the cell. What do you call the inside of the cell? What's that region called? Cytoplasm. By creating a vesicle. So this vesicle is going to be made up of the same stuff that the membrane is made up and the membrane of the organelles is made of. All right, so if I have a cell here, this cell wants to take in this little protein. So we're going to zoom in like I just did here. We're going to figure out exactly how this protein is going to make its way into the cell. Because there are things that want to hurt the cell. There are things the cell doesn't need. There's a way to bring in this necessary material, this necessary molecule. So we're going to zoom in. This is the plasma membrane. There's our protein. The cell membrane is going to go through an infolding. Do any of you happen to remember what this little infolding is known as? Do you remember from last time? Go ahead. Invagination. Good job. Emmy, right? All right. Okay. So that is a invagination. You can see that it's starting to pinch off. And before you know it, whoops. You've got yourself a vesicle. And this vesicle is going to go somewhere in the cell. It might go to the Golgi, the ER, the nucleus even. It's going to go to a certain destination within the cell. Now, to continue with endocytosis here, there's going to be 3 types of endocytosis we need to cover. And the first one is going to be called phagocytosis. Phage means to eat. Cyto means cell. Osis means process. So this process is all about taking in large macromolecules like a protein or a carb or a lipid and breaking it down to use their monomers. Now, I told you guys whenever we have an opportunity to go back to Unit 1, I like to take that opportunity because as we go further away from it, it's going to be harder for you to remember. So you don't need to write this down if you don't want to. But if you were to have a cell phagocytize a protein, what would that protein be broken down into? What are the monomers of a protein? Amino acids. What if that cell was to phagocytize a carbohydrate? What would that break down into? Emmy? Monosaccharides. Good. And how about if it were to break down a lipid, more specifically a triglyceride? 3 fatty acids and what? A glycerol. All right. Good job. Good, 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 good. Okay. So I have a question for you. The question is, how do cells know which proteins to get? Okay, so you got a cell. There's some proteins in its external environment. How does the cell which proteins it needs and which proteins it does not need? How is it able to differentiate things in the extracellular space that it needs and can bring into the cell versus things it doesn't need and wants to keep out? What is on a cell's surface that helps it to tell things apart? No, these things, what I'm thinking of is projecting from the plasma membrane. Yep, and glycoproteins and glycolipids. Right. Glycoproteins and glycolipids. Guys, these are going to be serving as receptors. They're able to differentiate what the cell does need and what the cell doesn't need. So it basically marks marks a vesicle for digestion or not. All right. Now to continue on with this about digestion and a vesicle breaking things down, I want to talk about something we did last week. It's going to be lysosomes. No, I just wrote in cursive. It's kind of weird, but it's because this is going to be a little different. This is associating one lesson to another. So lysosomes carry what type of enzymes? Do you remember what that H word was? These enzymes break things down into smaller molecules. Do you recall? Helicase? No, helicase is something that we haven't gotten to yet. Yeah. Hydrolytic. All right. So lysosomes, if you recall, are membranous sacs that contain hydrolytic enzymes. So why am I bringing this up? Here's why. A lysosome will attach to a vesicle that is intended for digestion. So let's say we have a vesicle here. And inside this vesicle is something that the cell doesn't need or the cell doesn't want. So we're going to say this is an unwanted, it could be an unwanted molecule. You know, it could be bacteria, which is an entire cell. This is just bottom line, the cell doesn't want it anymore. So here comes a lysosome. Inside the lysosome are going to be some digestive juices. These digestive juices are going to be the hydrolytic enzymes. The vesicle is going to fuse with the lysosome, and these digestive juices are going to go into that vesicle and basically break down whatever it is that's inside. What types of cells in your body would you expect to have a lot of lysosomes because of the lysosome's nature of digesting things in the body that are either unwanted or are a danger to the cell? What? No, the liver is what's responsible for cleaning out your blood. White blood cells. WBCs have many lysosomes. The proper name for a white blood cell is a leukocyte, by the way. Okay. Let's carry on. The next type of endocytosis we'll need to go over is going to be Pinocytosis. So this is something cell process. This is drinking cell process. In pinocytosis, you generally have vesicles are created that contain water and dissolved solutes. Okay, do any of you have a parent that drops you off at plants every day? Is there anybody that when you leave your car, is anybody that asks you who you are or they can have a form of identification? No. Most schools don't have that. You guys can just walk right in. Hopefully you're a student. We really don't. There's too many students. We don't have the resources to screen each one. Tom Brady could walk into this school and we wouldn't know who it is. Do you know who Tom Brady is? Marion, do you know what Tom Brady is?
Yeah.
Who?
He plays football.
Yeah. Do you know what he plays for?
Bucks.
Good job. Whoever whispered it to you. Way to
No
go.
one.
Really?
That was all me.
Yeah? What do the Bucks play?
When or
What do they play?
football?
Proud of you.
Thank you.
Yeah. So the reason I bring this up is pinocytosis is kind of like high school. Nobody's checking to see who you are. You could be good. You could be bad. It's really a crapshoot. There's no way to differentiate. Sometimes in pinocytosis, the cell might take in things that could hurt it. You don't know. Receptor-mediated endocytosis is like a bank on Wall Street. If you've never been in New York City, in South Manhattan, in downtown, there's an area called Wall Street. It's where all the stock markets are. It's one of the financial mega centers in the world. And there's banks that are not just American banks, like, Bank of America, Chase, Fifth Third Bank, all that SunTrust. There's places like Deutsche Bank, German banks. You got Swiss banks. You got French banks. You got banks from all over the world because it's a major hub for money, the stock market. And when I went down there, I remember seeing in front of all these banks that are from foreign countries, there were armed guards. I'm not talking some mall cop with a taser and a little Segway. I'm talking about guys wearing fatigues, military boots, little military hat, and he's got a machine gun. Because these places are on lockdown because they're very vulnerable, or not that they're very vulnerable, they're very important. There's a lot of very important business going inside of them. And so they need to be able to screen who's entering. If an employee wants to go in, they should have the credentials to get in. If I'm a tourist, I'm just walking through the stock market, I'm going to say, hey, let me into the Deutsche Bank. They're going to say, no, you have no business being here. Now go away. Receptor-mediated endocytosis is like pinocytosis in that you're taking in liquids, but it is going to take in specific things that it needs because in receptor-mediated endocytosis, it uses receptors to differentiate what it does and what it does not need. If I'm standing by my door and I see Zespina come in, she's allowed to come in. She's my student. But if I see Marcus Williams come in, I don't have a student named Marcus Williams, so I'm not going to let Marcus into my classroom. He has no business being here. So it's kind of like you have a receptor-mediated endocytosis in this room. I check to see who should and should not be here. So in receptor-mediated endocytosis, you have specific receptors protect molecules in solution and trigger the formation of a vesicle. So in these, the vesicle is going to be intentionally formed around a necessary molecule or solvent. Okay? And now we're at the conclusion for today, guys. What is the opposite of endocytosis? Exocytosis. Marion, what does exo stand for?
Exit.
Very good. Let's see. Hannah, what does cyto mean?
Cell.
Uh-huh. And Annabelle, what does osis mean?
Process.
Very good. So this is exit, cell, process. We have the transport of molecules outside of the cell by fusing a vesicle with the plasma membrane. So we're going to zoom in here. This red line is our plasma membrane of a cell. Here we have our vesicle. And here we have our protein. Could be a protein, could be a carb, it could be whatever. Bottom line is it's going to leave the cell. Orson. Lauren Clark. Yep. Signing out. We're almost done if you want to hang around for one more minute. All right. So this vesicle is going to fuse with the membrane, very similar to how it's going to be the reverse of endocytosis. Why is the vesicle able to fuse with the membrane so fluidly?
Is this only happening with non-polar molecules?
The vesicle is, no, it's happening with very large molecules.
So how is like the membrane forming around the molecule when it's outside of the cell for endocytosis? If it's like the molecules here and plasma membranes here, what's allowing it to go through?
The receptors. The receptors are starting to the receptors trigger the membrane to invaginate to allow the molecule to come into the membrane to form a vesicle.
So like even though it's not non-polar.
It doesn't. It's really not to do with polar or non-polar. It's about how big the molecule is. These molecules are far too big to just pass through the membrane, so they need. That's why this is called bulk transport, because they're so big. You need to form an actual vesicle around them, and you can't expect them to just travel through a port. If I wanted to bring in an elephant into this classroom, an elephant's not going to fit through that door. It's far too big. Imagine that door is a protein channel. This wall is the plasma membrane of a cell, and that door is a protein channel. An elephant is not going to pass through that. So you're going to need something a lot bigger in order to encase that elephant to get it in here. Maybe we have to, what if this wall, imagine this, what if this wall could wrap around the elephant and bring it in? Instead of the elephant just trying to pass through that door, which is serving as a channel. That's the difference between the typical sodium potassium pumps or the active transport processes or passive transport processes we've done. So this isn't really so much a matter of polarity, it's a matter of size. These molecules are just really, really big. Okay, and then finally, didn't mean to draw that. Finally, the protein or the molecule, whatever it is expelled from the cell. Now, don't you be thinking that this is something that the cell is trying to get rid of as if it were garbage. It's not garbage. When you seniors, you graduate from plant and you leave plant forever, it's not because you're garbage and we're trying to get rid of you. You're just moving on to the next step of your lives. This protein is moving on to the next step of what it's meant to do. It's already accomplished the part where it needs to leave the cell. Maybe it needs to go do something in the extracellular space. Maybe it's needed by another cell somewhere far away and it needs to travel to it. So this is not necessarily the cell just getting rid of trash. That is an option, but it's not the only option. Okay? All right, that is it for this lesson.